Hello, class. So you have had the bone anatomy, the bone lecture, the muscle anatomy, and now the muscle lecture. So this next test coming is bone and muscle. And there's a lot of anatomy. I'm checking exactly when your exam is going to be. So the quiz is November the 2nd, and the exam is November the 4th. So you want to make sure that you are between now and then getting through that anatomy, actually putting a lot of time in. You haven't had this much anatomy yet. And so you need to put some time in going over those things. Anything you have questions about, you need to email me so we can chat to discuss them. The notes will be up for both the bone and the muscle. Um, the models are already up on the discussion board for bones and muscle. The study guide is already up for this exam. So anything that you have questions about as we're going through, jot them down, email me so we can answer them. So I'm going to turn me off and we're going to get into this information. Okay, so muscle is specialized tissue for contracting or shortening. And so it provides movement. And we have three different types of muscle in the body: cardiac muscle, smooth muscle, and skeletal muscle. And much of this will be about skeletal muscle. We will touch on the other two, but we're mainly going to talk about skeletal muscle. So, skeletal muscle allows for us to actually move from one location to the next, right? Locomotion. It allows for us to manipulate our environment, right? If I want to pick up a pencil, if I want to make a facial expression, those kinds of things. Muscles attach across the bones and they help maintain posture. Stabilize joints, for example, the shoulder joint. There are four muscles there involved in creating their rotator cuff. Because if you think about the head of the humerus, it doesn't fit snugly and tightly in the glenoid cavity. The glenoid cavity is really shallow. And so you need muscles and the tendons of muscles to support that joint. Whereas the head of the femur fits very snugly in the acetabulum. And then muscle contraction helps to generate heat. Okay. So when we talk about all of the types of muscle, they are all excitable. So all cells have what's referred to as a resting membrane potential, the charge across the membrane, with sodium at a higher concentration outside and potassium at a higher concentration inside. And then excitable cells will have the movement of that sodium and potassium to stimulate and have the cells recover. We'll see that for muscle cells and nerve cells. Other cells have that resting membrane potential, but they aren't excitable the way muscle cells and nerve cells are. Sometimes you might see the word irritable related to that. Muscle cells shorten or contract. They extend, they bounce back to that original resting position. Some of them contract by extending. The gastrocnemius is one. Um, and that elasticity is that ability to bounce back and go back to that original position. So when we compare muscle cells to all other cells, they are elongated. We already said they contract, and they have the contractile proteins actin and myosin. I'm just going to put a little side note here for actin and myosin. Okay. So the actin and myosin are the contractile proteins, and they actually alternate. The thin and thick filaments alternate, and they create the stripes or the striations we see in skeletal muscle and cardiac muscle. Smooth muscle has actin-myosin, but not arranged in the same fashion. That's why we don't see any stripes or striations there. Skeletal muscle typically has one nerve, one artery, one major vein, sometimes more than one vein. So if you see something that says the innervation, that means what nerve is supplying that muscle. So for example, you'll have a femoral nerve, a femoral artery, a femoral vein. Okay. And then along with the femoral vein, there may be some other veins that would drain the surrounding area. So arteries are vessels that supply oxygenated and nutrient-rich blood to the muscle, and veins drain that oxygen deprived or oxygen poor blood from the muscle. Okay. And then I ask here, what is a motor unit? A motor unit is a motor neuron, and a neuron is a nerve fiber, a motor neuron, and all of the muscle fibers it touches. So you will have one nerve that has all these little tentacles called axon terminals, and then at the end of the terminals are referred to as terminal buttons. And I think we looked at that when we looked at tissue. We looked at the neuron, and then those terminal buttons will touch muscle fibers, and so that's going to be important for stimulating more muscle fibers to contract. So for skeletal muscle in particular, there are no gap junctions. And we're going to talk about that table again. You had a table differentiating the types of muscle when we talked about tissue. We're going to talk about that table again. But smooth muscle and cardiac muscle had gap junctions where they send chemical signals from one cell to the next to say, okay, let's contract. Skeletal muscle doesn't have that. Skeletal muscle has motor units where you have nerves innervating these fibers. And the more muscles I need to contract, the more motor units I'm going to recruit. So there's a process called recruitment. And so if I need to pick up a pencil, I don't need to activate a whole lot of motor units, right? If I want to pick up my book bag, well, now I got to activate more motor units. If I want to pick up a boulder, right? So now I got to activate more, right? And so that's what we're talking about there. Okay, so this is the table that you had before. You need to know it. Hopefully you remember it from before when you differentiate the different types of muscles. So the only one that is voluntary, meaning you have control over it, is skeletal. You have no control over your heart contracting. You have no control over your blood pressure or the stuff moving through your gut or any of those kinds of things. The most rapid is skeletal muscle. And when I say the most rapid, we're talking about a muscle twitch, the stimulation, the contraction, and the recovery. They're all fast when we think about actual time. We're talking about fractions of a second. But when we compare the three, skeletal is the most rapid, then cardiac, and then smooth. We already said that the cardiac and smooth have gap junctions. And the cardiac also has intercalated discs, points of connection at the those heart muscle cells, and there's also branching there. The skeletal muscle is created to be arranged in bundles. So when we name a muscle belly, that is a bundle. The heart is a closed-ended tube, and smooth muscle is arranged in sheets. And sometimes then those sheets curve around and make a tube, like the GI tract or like the middle layer and blood vessels. Skeletal muscle has a lot of nuclei, and they're pushed to the edge. And cardiac has 1 to 2 centrally located nuclei. Smooth muscle has 1. I'm going to zoom in on this picture at the bottom. This should look familiar. So here is my skeletal muscle. You see those stripes going up and down, not across. So let me show you exactly what I'm talking about. These lines here, you see these lines going up and down like that, light and dark bands. Those are my striations. This is one muscle fiber. This is another muscle fiber. This is another muscle fiber. And then these are the nuclei pushed to the edge here. Okay, so these muscle fibers are lying together next to each other, packed tightly. Okay, and then the stripes are running like this. Those are the striations. Okay, and then on the little illustration down here, you see the stripes or striations, and you see these nuclei on the edge. Here's cardiac muscle, 1 to 2 centrally located nuclei. You got the intercalated discs, and you can see the striations or stripes there, still running in that same direction. You see the branching that cardiac muscle has. The other ones don't have that. And then we have smooth muscle here. And these fibers look more like this, and they just lie end to end together here until they are packed tightly together. So you'd have one here, and then one here, and then one here, right? And that's how they're arranged. And you have a nucleus inside. Okay, no striations here. Okay. So when we talk about skeletal muscle, it's arranged in a very specific way. Each muscle cell or fiber is wrapped in a connective tissue covering called endomysium. So each fiber, each cell and fiber is interchangeable here, is wrapped with endomysium, and then the fibers or cells are bundled together, and they're wrapped with perimysium. And each of those bundles is called a fascicle. So if you've ever had stringy roast beef, the stringy parts, those are fascicles. And then the fascicles are bundled together and wrapped with epimysium. And the epimysium is going to lead to the tendon. So we see that here. Okay. Let's look at this up close. So we have our endomysium surrounding each muscle fiber, right? So here's a muscle fiber, and that little white around it is the endomysium. Then we have them bundled together, and they're wrapped with perimysium. So this is a fascicle. And then we got a bunch of fascicles, and the fascicles are wrapped with the epimysium, right? And then that leads to the tendon. Okay, so we got endomysium, and then we have perimysium, and this is also perimysium wrapped around the fascicles. The fascicles are bundled and then wrapped with epimysium, and epimysium leads to the tendon. And that is specifically the way the skeletal muscle is structured. Now, cardiac muscle has some connective tissue around it too, but it's not as regularly arranged as the muscle bellies of skeletal muscle. Okay. So the epimysium is going to lead to a tendon or an aponeurosis. And aponeurosis is like that gala aponeurotica across the skull, that flat band of connective tissue connecting the occipital muscle and the frontalis muscle. We talked about the nerves and the blood vessels, so we're good on that. Cardiac muscle only makes up the heart. Again, there's connective tissue fibers interwoven there. It actually creates what's referred to as a fiber skeleton in the heart. And I don't have that stated here, but you can write that down, that there's a fiber skeleton that is created because of the connective tissue fibers in the heart. But not regularly arranged like we saw in skeletal muscle. The cardiac fibers are arranged spirally, so you see that here, right? And so that is so that you get the maximal amount of blood expelled during contraction. So you get kind of a ringing action, okay? And so that's why the fibers are arranged spirally. You can see that here. And then this is showing you the lower chambers of the heart um, contracted and relaxed. Smooth muscle is also referred to as visceral muscle. Viscera is a term that means organs. So smooth muscle makes up hollow organs like the stomach, the um, whole GI tract, all the intestine, the esophagus. It makes up the bladder. It makes up the uterus. It makes up the gallbladder. It makes up blood vessels, the middle layer of blood vessels, particularly arteries. Lots of smooth muscle there. And so a lot of the activities that smooth muscle is responsible for are kind of taken for granted. So a lot of the textbooks will say they have housekeeping activities. Housekeeping activities makes it seem like these things are not important. Super important responsibilities of smooth muscle. Of course, we can appreciate the super important responsibility of the heart, right? It stops beating, you stop living. We appreciate the super important responsibilities of skeletal muscle for you being able to get up and move around and manipulate your environment and so on. We kind of take for granted smooth muscle. Oh, it has housekeeping activities. Listen, smooth muscle is keeping your blood pressure regulated. Those small arterioles, tiny arteries um, in your body, that smooth muscle activity there helps to keep your blood pressure regulated. If you eat that burger and your smooth muscle in your gut is not working right and that burger material stops moving, we got a problem. Or if it moves too fast, we got a different problem, right? So, smooth muscle activity, while they call it housekeeping activity, is really important. Okay. And so, when we say all the cells are elongated, skeletal muscle cells can be super long. Like you remember the sartorius, that strap like muscle that goes across the thigh. Okay, all of those cells go the full length of that muscle. Okay, smooth muscle cells are not as long as that, but they're not like round um, cells like when we draw a generalized cell. They're elongated. All of them are, but a lot of those skeletal muscle fibers are really long. Okay, so this is showing the muscle relaxed and contracted. Okay, so here is an up close of some cardiac muscle and some smooth muscle. Let's zoom in. So this is our cardiac muscle. You see the stripes or striations, you see the nuclei, you see the branching, and then the illustration here up close shows you the gap junctions, which are these little connections that also have little tunnels for chemical signaling. And you have these little plaques called desmosomes at the intercalated discs to keep the cells attached. And then here you can see the branching and you can see many mitochondria in there, right, for um, providing energy or ATP. This blue and gray here, I refer to it as the T-tubule system. It houses calcium. That's going to be important for contraction for cardiac muscle as well as skeletal muscle. And we'll talk about that more with skeletal muscle. That calcium is going to be key for contraction. And then the red and the blue here, and I'm going to zoom in a little bit more on that. The red and the blue represent the alternating bands of actin and myosin. So the blue on here represents actin. The red on here represents myosin. Myosin is thicker. And so these alternating bands create these light areas here and darker areas here, which are our stripes or striations. And then here, smooth muscle, these fibers are bundled together. And you see they'll bundle together here, and the nuclei are centrally located. Okay, so no stripes here. So we know that muscle is going to allow us to have movement, mobility of body parts, locomotion, movement of content for the heart and the smooth muscle. All right, so we're going to move content through the body. Muscle helps in maintaining posture, providing tone. What do we mean by tone? Tone is where you have this semi-state of contraction of muscle. So as long as the person is alive and there's some calcium available, you're going to have some muscle tone. So for example, when we talk about the gut, the intestine, when the person is alive and we look at the stretch out that intestine, it can be, about 13 feet. And if we look at it in the cadaver and the individual is not living and you stretch it out, it's about 20 feet. Okay. So that partial state of contraction is tone. We already said that skeletal muscle in particular can help stabilizing in stabilizing joints, and muscle contraction generates heat. We talked about that when we talked about the skin and that erector pili muscle in the skin. This is why you get goosebumps, right? You go from one room to the next and the room is cold and then you get goosebumps, right? Those muscles are contracting to generate heat. Here, when we talk about muscle contraction generating heat involuntarily, if you're standing at the bus stop and it's 10 degrees out, you might start shivering or you might have your teeth chattering, right? Your muscles are involuntarily contracting in an attempt to generate heat. And you can appreciate that when you are purposefully contracting muscle, like working out, you generate heat, right? The muscle activity generates heat. So now, this is the structure of skeletal muscle. So this looks a little bit similar to what we saw for smooth muscle. I'm just going to zoom in on this first, and then we'll go back to the note part. So this whole thing is the muscle fiber or the cell, and you have these fibrils in there, and these are areas that are having the arrangement of actin and myosin bundled together. You have a lot of mitochondria scattered throughout, and then this blue and yellow, I call it the T-tubule system. It has these tubules and it has a sarcoplasmic reticulum, which is a specialized endoplasmic reticulum that houses calcium. And we need the calcium for muscle contraction. And we'll talk about that in a minute. Okay. And we'll define sarcomere in a minute as well. But that is what this looks like microscopically. So let's define a couple things. So we got the sarcolemma, which is the cell membrane. We got the sarcoplasm, which is the cytoplasm inside the cell, right? So these things are things that we're familiar with just for any cell in general, but we have special names in muscle. So sarcolemma, sarcoplasm. Then we have something called myoglobin. Myoglobin is a red pigment and it stores some oxygen. And that's important because oxygen is required for the reactions that produce ATP. And so we need ATP because it is energy, and that's how our cells are going to do work. So we have some myoglobin in here that gives muscle its red color, and it has oxygen attached, and that oxygen is there to help in the production of ATP. Okay. Myofibrils, those are the long fibers inside, and those are created by that actin and myosin. So then the thin filament we already said is actin. The actin has binding sites for myosin heads. Those binding sites are typically covered when the cell is at rest. So they have some proteins covering them. There's a long spiraling protein surrounding the actin called tropomyosin. And attached to the tropomyosin is another protein called troponin. Troponin is a small little protein, and it has three little parts to it. One that's going to bind to actin, one that binds to the tropomyosin, and the other one that is available for calcium. That's going to be important. Myosin has an arrangement where you have something that kind of, let me do this. Let me make it a little better by doing this. So you have something that looks like this, right, for myosin. And you get a bunch of these bundled together. So you have this tail and two heads. And so if we get a bunch of them bundled together, we're going to end up with a thick filament. Right, so we get a bunch of them together. And then you see this filament is thick, right? And then you have these myosin heads that want to attach to the actin, okay? But the actin binding sites for those heads are typically covered at rest, and we'll talk about that in a second. You have some elastic filaments, and they're made of a protein called titin, and they help to keep the muscle able to recoil. Let's zoom in on this picture at the bottom. Now, this is called a sarcomere. Sarcomere. A sarcomere is one contractile unit of striated muscle. That on the side. Okay. Not added, it goes from Z line to Z line. Okay. So let me show you exactly what I mean here. I'm going to zoom in even a little bit more. I'm going to give you a second to write that down. I'm going to zoom in even a little bit more. Okay, so let's zoom in a little bit more. Okay, so we see our myosin here. This is myosin. It's thick, and you see these myosin heads. Okay, and then out here we have our actin. It's thin, in comparison. And you see we alternate thin and thick filaments, thin and thick filaments, thin and thick filaments. Now, we have some designated areas here from here to here, where we have alternating actin and myosin. This is the A band, and it's probably typed down there, but I don't want to move this at this point. This is the A band. Okay, so the A band is all of this area in here where I have both actin and myosin. Okay, in the center of the A band where I don't have any actin, this is the H zone. And then over here, and there's a blue line around it, we have the I band. We have it over here too, where there is no myosin. That is the I band. So that would be a lighter area. The A band is going to be darker. The I band is going to be lighter. And then in the middle of our I band is what is referred to as the Z line. The Z line. So there is some titin or this elastic kind of fibers here. Don't worry about M line. Don't worry. I'm going to scratch out. As a matter of fact, let's do it this way. Some stuff I don't even want you to worry about. We're going to do this. You don't need to worry about any of those things that I have crossed out. This is the stuff you need to worry about. Okay, the A band, right, that has actin and myosin. The H zone in the center where there is no actin. And over here, the I band where we have no myosin. And in the center of the I band is the Z line. This is a sarcomere, one contractile unit of striated muscle. Okay. So this is what that skeletal muscle looks like. So let's I got two separate ideas here. So here's my skeletal muscle fiber. This is a cell. You can see a lot of nuclei pushed to the edge here, right? And then you see the striations, light and dark and light and dark. And that's because of the arrangement of these fibrils inside the actin and myosin. So now we look at that up close. We see the cell membrane is the sarcolemma. Here we have this T-tubule system in here with the tubule and the sarcoplasmic reticulum. We got mitochondria in here. We can see our alternating bands of myosin and actin creating these stripes or striations, right? And if we blow that up, then that's what we see here, right? So we got our A band that has actin and myosin in it. In the center of that, we have our H zone, right? No actin there. Over here, we got our I bands, right? In the center of the I band is the Z line. So from Z line to Z line is our sarcomere. Okay, so from Z line to Z line is our sarcomere. And you see the alternating bands of actin and myosin here. Okay, let's look at the other picture. Same thing. This one is a little bit blurry. They are pulling the sarcolemma off here, and you can see the myofibrils. Here you can see our sarcomeres. We got our alternating bands of actin and myosin here. This is our Z line, right? So this is my A band, in the middle of it, my H zone, my Z line in the middle of my I band. Okay, so from Z line to Z line is one contractile unit or one sarcomere. And if we look at that up close, here is our myosin. The red represents myosin. I'm going to type in here. That's myosin. And then this is actin. The little curly yellow is the titin. But you can see here, we go from Z line to Z line, or they call it the Z disc here. That's my sarcomere. Right. Here is my A band, where I have both actin and myosin alternating. In the center of that is my H zone, right, where I have no actin. Over here is my I band, where I have no myosin. Okay. So, how would I describe my A band? It's a dark band. It's a link of the myofibril, and I'm going to add to that because the A band contains both actin and myosin. I band is in on either end of the A band. These are light bands, and they contain only actin. When I say only actin, there's some titin there, but the titin doesn't contribute to making up the striations. The Z line is in the middle of the I band. And the H zone is the lighter area in the middle of the A band, and the H zone contains only myosin. And I'll let you write that, then I'm going to zoom in on this picture. Zoom in. So this is my sarcomere. The dark blue on here is the myosin. The red is the actin. And so you have your A band here. In the center of it is the H zone here. Over here is the I band. And in the center of it is the Z line. Right? I band, Z line. Okay, so from Z line to Z line is the sarcomere. If we blow this up and we look at this actin and myosin, so this is our actin. I'm going to zoom in a little bit more. It's not a super clear picture, but this band of tissue swirling around here, that's my tropomyosin. And then attached to the tropomyosin, these little areas here, that's my troponin. Okay, so let me label that. Okay. Now, if you look at the actin itself, it looks like little red balls all joined together. And the little red balls have these yellow spots on them, if you can see that. Those are the binding sites, right? You see those? But they're covered up by this tropomyosin. That's going to be important when we talk about this. Okay, so just keep that in mind. Here is my myosin. We talked about it having the tail and the two heads on the end, and they bundle together to create these thick filaments. And then you have some elastic kind of connective tissue here at the end so it can recoil. Okay. And so the actin and myosin alternate and create the stripes or striations. Okay, so now how does this process work to create a contraction or the shortening? You might hear the term cross bridge activity. You might see the term power stroke. I don't really use those terms. I just talk about the process. And so I have some steps here, and it's not important that you memorize this in step 1 is this, and step 2 is this, and step 3 is this. What's important is that you understand the sequence with which things happen for this process. What happens is students try to memorize steps with a certain number of steps. I can discuss this in 3 steps. I can discuss this in 12 steps. So it's not the number of steps that's important. It's the process that's important. And I'm going to flip to another page to describe this, and then I'll go back to that and let you view it from there. So I'm going to discuss what happens. Okay, so the first thing that happens is a motor neuron is stimulated, right? Because we talked about the motor unit. When that motor neuron is stimulated, it releases a chemical at the end. The little terminal buttons have vesicles in them. Remember, we talked about vesicles inside of cells that contain things. Well, these things that are contained within the vesicles are called neurotransmitters. So the motor neuron is stimulated and it releases neurotransmitter. This particular neurotransmitter is called acetylcholine. Okay. The acetylcholine is going to trigger the release of calcium from that T-tubule system. Now, there's some electrical stuff that happens here as the nerve is stimulated with sodium and potassium. I'm not going to add that here because it's already a lot to talk about the contraction itself. We'll talk about what happens with sodium and potassium when we talk about the nerve, and then I will backtrack to relate that to muscle because it's very similar. But here, I want you to know that the muscle gets stimulated because of that acetylcholine, and it's going to cause the release of calcium from the T-tubule system. Okay. When that calcium comes in, it's going to bind to that troponin. That was that little small piece. attached to the tropomyosin. Tropomyosin was that long, thin part, overactive. The long, thin part is covering up the binding sites. Once calcium comes in, it binds to the troponin. When it binds to the troponin, that causes the tropomyosin to shift, and now the binding sites are exposed. When the binding sites are available, myosin heads will attach to them. And that causes the contraction. So when the myosin heads attach, they bend, and that causes the contraction. Now, this contraction is not one big fell swoop. It's kind of like a tug of war. So we have some myosin heads that will attach and bend, while other ones are reaching out, grabbing further and bending. So it's kind of like a tug of war. Not everybody on one side of the rope lets go at one time. So you got some people pulling and other people holding, and then they switch until you keep pulling that rope closer and closer to you, right? So that's what they do here. This should say no S, just myosin here. I don't know if I can go back up and fix it. Yeah, I can. Yes. Good. Now, when that happens, you get the contraction, and when you get a full contraction, you're going to lose the I band and the H zone. You need the ATP to put the myosin heads back in their original place, either to do it again or to fully relax. Now, the way that I describe this in the notes, and I'm going to go back to it, probably a whole lot more information, maybe more steps, maybe fewer steps. I don't know. But this is a process. So the first thing that happens is that you get a nerve stimulated. That nerve releases a neurotransmitter. In this case, this is acetylcholine. Acetylcholine is going to stimulate the release of calcium from the T-tubule system. It's going to excite the muscle cell. The muscle cell is going to release the calcium from the T-tubule system. The calcium is then going to attach to troponin. When it attaches to troponin, then the tropomyosin shifts out of the way. The binding sites are now open. When the binding sites come open, the myosin heads will attach, and then they bend, and that causes the contraction. Again, this is like a tug of war. They don't bend all at once. It's a stepwise fashion of contracting. And then the sarcomere will fully contract if that is what's called for. And then once you have the full contraction, you lose the H zone and the I band. And then you need ATP to put the myosin heads back as you're doing the stepwise process or when you want to fully relax. Now, I'm going to draw an illustration of how I demonstrate the sliding filament mechanism. That's what this is called. And I'm also going to post a video because that might be helpful to actually see it in action. So look for that video as well. So let me go to another page. And I'm going to draw my own little picture for actinomycin. Yeah, I don't want it that big. Second, it's going to take me. You know what? Maybe I won't have you sit for all of this. Okay, I didn't want to have you wasting time watching me draw all of this. Okay, so this is myosin, the thick filaments. The thin filaments are actin. Okay, now you see the myosin has these heads that want to bind to these little blue cups on actin. But there's a black squiggly line there that's covering that up. That is a tropomyosin. Okay, so now for the record, from here to here, right, that's my A band. In the middle of that is my H zone. Over here, from here to here, right, and over here from here to here is my I band, and in the middle of that is my Z line. I'm not going to mark that on here. I'm actually going to erase all those lines, but this is a sarcomere. Okay, so this is relaxed. Let's put that down here. Okay. So the myosin heads want to attach, but they can't because my tropomyosin is covering those binding sites. Now, we're going to get a stimulation, and we're going to get some calcium rushing in, and things are going to change. Okay, so this little purple dot is going to represent my calcium. Okay, now let's draw our contracted sarcomere. Okay, so now we have our contracted sarcomere. And you would not believe that I deleted that and had to draw everything all over again. Okay. So our contracted sarcomere, you see the calcium came in, it binds to the tropomyosin. Now the tropomyosin is out of the way. The myosin heads can attach and bend. So you see here now we don't have any areas where we don't have both actin and myosin. We lost our H zone and our I band. Okay. This is my relaxed sarcomere. You can see the clear A band, H zone, I band, all of that. My binding sites are covered with the protein called tropomyosin. Calcium comes in, attaches to troponin. That shifts the tropomyosin out of the way. The binding sites are available. The myosin heads attach and bend, and that's our contraction. And during our contraction, we lose the H zone and the I band. So I added some other myosin from the other sarcomeres nearby because all of them shorten together. And that's why you have myosin over on the sides that you can see here that you couldn't see on the relaxed one because those sarcomeres shorten too. Okay. So let's go back and look at the steps that I put in here. Okay. So we got the muscle receiving information from the motor neuron, a stimulation, right? The release of the neurotransmitter acetylcholine. That acetylcholine causes something called depolarization. And depolarization and repolarization are the movement of sodium potassium. So depolarization is when sodium is going to move in, and then at the end for relaxation, potassium will move out. We won't talk about that a lot at this point. I asked a question about acetylcholine esterase. Acetylcholine esterase is going to degrade the acetylcholine that's left where the nerve and the muscle meet, so that we don't keep stimulating the muscle when the process is over. But don't worry about that at this point. Let's worry about the steps. So first, we stimulate a neuron, a motor neuron, release neurotransmitter. The neurotransmitter is going to stimulate our muscle cell and cause a process called depolarization. That stimulation is going to move down the length of the nerve. That process is called propagation. Move down the length of the muscle. That process is called propagation. You don't really have to worry about depolarization, repolarization, propagation with full understanding at this point. We'll talk about it with the nerve. Okay. And then that stimulation is going to cause the release of calcium from that T-tubule system. That calcium is going to go in and attach to troponin, and that's going to cause the tropomyosin to shift out of the way, and the binding sites are going to be open. The myosin heads are going to attach to those binding sites and bend inward. That causes the contraction. That's the cross bridge activity. When calcium stops, the tropomyosin will go back to its original state. Okay, and you can put that or not put that, but ATP is required to put the myosin heads back into their original position, or recock the myosin heads to continue the contraction or for relaxation. Okay. So here's a picture of that. So this represents the myosin. This represents the actin. So let's start at the top where number 1 is. And so these, let's see. It looks like this is just talking about our ATP. So let's zoom in on this a bit. So this is my myosin here. This is myosin. Right, and this is actin. You see the little yellow circles, right? That's the actin, and the little black dots on them are the binding sites. This brown line here, that's tropomyosin. And the little blue part attached to it, that's troponin. The little purple is calcium. So calcium has bound to the troponin and shifted the tropomyosin out of the way, and that's why you can see the binding sites. And so the myosin heads want to attach to these binding sites. Okay, and so they do, and then they bend, right? Let's zoom back out. So they attach, they bend, that causes my contraction. At the end of that process, I need ATP to put my myosin heads back to their original spot here so that they can do it again. So they can grab another binding site and bend again. So this is why we need the ATP. Okay. Let's see. Okay, that was for me to draw it. I already drew it. Where do we get this ATP from? Couple of different sources. Direct phosphorylation and anaerobic glycolysis. Those are for short-lived amounts of ATP. Nothing that's going to be sustaining. Okay. And the one that causes us to get the most ATP is aerobic respiration. And aerobic respiration occurs in the mitochondria, where the mitochondria are producing ATP, lots of ATP in the presence of oxygen. These two steps don't have any oxygen, and that's why we can't get a lot of ATP produced. But here, where we have aerobic implies in the presence of oxygen, we get lots of ATP produced, right? And so that becomes important. So that's where we get the bulk of it. This is only referred to as giving minute amounts of ATP. Okay. So these are the steps involved in the production of ATP by aerobic respiration or cellular respiration. I want you to know the steps in order and what we start with, what we end with, okay, and where in the cell they occur. Now, you could learn this in a whole lot more depth. We won't do that here. I tend to go over it again in 153 with digestion, because that's the whole point of us eating, is to get the stuff we need to make energy, right? The molecules we need. So I talk about it there. But because we mentioned it for the purpose of creating ATP for muscle contraction, that's why we go over here. So the 4 steps: glycolysis, acetyl-CoA formation, citric acid cycle or Krebs cycle, and then the electron transport chain. These 3 steps occur in the mitochondria and require oxygen. So I'm just going to make a note of that right here. So if you need to zoom in on this, hit those 4 arrows pointing out and it'll get bigger and you can see it better. So these 3 steps require oxygen and they occur in the mitochondria in the cell. This step occurs in the cytoplasm and does not require oxygen. Okay. So the first step is glycolysis. Now, again, I want you to know what we start with and what we end with. What's the name of the steps in order? Glycolysis, acetyl-CoA formation, citric acid cycle or Krebs cycle, electron transport chain. Where they occur: glycolysis in the cytoplasm, the other 3 in the mitochondria. Glycolysis doesn't require oxygen, the other 3 do. Okay, so now, what do we start with glycolysis? We start with glucose. And so I say sugar here, but I'm going to be very specific here and say glucose because that's important to know that we're talking about glucose. Not even sure why I said sugar. Other sugars will break down into glucose, but we start with glucose. And we end up creating a molecule called pyruvate or pyruvic acid. We get a little bit of ATP and a little bit of NADH here. Now, this pyruvate goes into our next step. So our next step always starts with what we ended the last step with. So we start here with pyruvate. We end up with acetyl-CoA. We get a little bit of NADH formed here as well. NADH and FADH are electron carriers. And I'm just going to make a note of that down here. So go back. Glycolysis, we started with glucose. We end with pyruvic acid, a little bit of ATP, a little bit of NADH. Then we go to our next step, which is acetyl-CoA formation. We start with that pyruvate that we ended the last step with. We form acetyl-CoA, a little bit of NADH. Then we go to our next step. What goes into our next step? The acetyl-CoA. And then we end up producing a lot of NADH and some FADH. And a little bit of ATP. We get a little ATP, lots of NADH, and some FADH. Now, once we do this glycolysis step, there are 2 pyruvate molecules, so both of them go through these cycles. Then our next step, we take this NADH and FADH. So we start with NADH and FADH. Those feed into the electron transport chain. So that's what we start with. And we end up with lots of ATP here. And here where I say oxygen is the final electron acceptor, that allows us to produce a little water. The electron, what has 1 electron? Hydrogen. So oxygen receives that. We end up with a little water. Okay. So the first step is glycolysis. It occurs in the cytoplasm. It doesn't require any oxygen. We start with glucose. We end with pyruvate, a little bit of ATP, a little bit of NADH. Okay, so then we take that pyruvate, we go to our next step, which is acetyl-CoA formation. We start with our pyruvate, we end with acetyl-CoA, a little bit of NADH. Okay, then that acetyl-CoA goes to the next step, which is the Krebs cycle. We start with acetyl-CoA, we end up with a little ATP, but lots of NADH and some FADH. The NADH and FADH from every place we made it goes into our electron transport chain. That occurs in the mitochondria. It requires oxygen. And here's where we make lots of ATP. I'm not concerned about the numbers of ATP in other classes. You'll learn it and they'll talk about each step and all of the chemical reactions and where that ATP is produced and how many molecules at each location. We're not concerned about that at this point. Okay, let's go through it again. So we start with glucose, which is sugar, but we want to just say glucose. Matter of fact, let me type it. Just that's not clear. So we start with glucose. We end with pyruvate or pyruvic acid. And this is our starting point. This is our ending point. We also get a little bit of ATP and NADH. Our next step, we take that pyruvic acid or pyruvate and we form acetyl-CoA. In this process, we get a little bit of NADH. That acetyl-CoA goes into our next step, the Krebs cycle, and it allows us to produce a lot of NADH and some FADH. We also get a little bit of ATP here. The NADH and FADH from every source we made it goes into the electron transport chain. So let me just make a note here that this starts with and so it starts with the NADH and FADH, and then we end with lots of ATP here. So this is our main source for producing ATP. Okay, so this is cellular respiration or aerobic respiration. So here we have glycolysis out in the cytoplasm. We start with glucose, we end with pyruvate. Then we have our next step, which is acetyl-CoA formation. We start with pyruvate, we end with acetyl-CoA. Our next step is the Krebs cycle or the citric acid cycle. We start with that acetyl-CoA. We end up with lots of NADH and FADH, a little bit of ATP. Then we go to our electron transport chain, and we start with that NADH and FADH, and we end up with a lot of ATP. Again, I'm not concerned about the numbers. Just know that that's the main source of ATP production. And the yellow here represents the mitochondria. Okay. We're not going to even worry about that. Okay. Types of muscle contraction. Isotonic contraction versus isometric contraction. So we talked about contraction, right? We talked about that sliding filament mechanism, that actin and myosin and those filaments sliding across each other. That's what we mean by sliding filament mechanism. So when we have an isotonic contraction, that's what happens. We actually get the shortening of the muscle as those filaments slide across each other. We can have a concentric contraction. That muscle shortens and does work. Eccentric contraction, the muscle contracts as it lengthens. This is the example of the gastrocnemius. Most muscles are concentric contraction. Isometric contraction is that there's no shortening of the muscle. There's just a building of tension, a development of tension. And so what happens is you stimulate the muscle, you get the acetylcholine release, you get the calcium release, you get the myosin heads attached there, but we don't actually get the sliding filament mechanism. So an example of that would be like if you were trying to push down a wall. You just develop a lot of tension. You can't push the wall down. They're showing the guy here. I don't know if this is called a plank that he's doing, but he's not really contracting the muscles in the sense that the filaments are sliding back and forth, but he's developing a tension there. Okay, that's isometric contraction. Some definitions here. We mentioned muscle twitch before. This is where we get a muscle being stimulated and then there's a brief response with a recovery. So a muscle twitch is the stimulation, the contraction, followed by the recovery. Summation is when you get an additional contraction before the muscle actually has time to fully relax. And I think I have some pictures of that in here. We mentioned recruitment before. Recruitment is where you have a number of muscle cells stimulated by motor units as needed, right? So if I pick up a pencil, I only need to stimulate a very few number of motor units. But if I pick up my bag, I got to stimulate more. If I pick up my dog who weighs 70 some pounds, I got to stimulate more, right? So that's recruitment. Tetanus is if I hold the stimulus steady and the muscle never gets to relax, there's going to be a sustained contraction for a short period of time, and that contraction is going to be weak because you're going to give out after time. And then you have fatigue, and fatigue is when you are unable to contract or sustain a contraction because of an ATP deficit. And we defined tone earlier, the slight state of muscle contraction or that partial state of muscle contraction, even during relaxation. So the muscle is always slightly contracted. So here's what we're talking about a muscle twitch. So now you got a stimulus. There's a short delay here called a latent period. Let's zoom in on this little picture here. So we have a stimulus, and then there's a very short delay here before the muscle actually reacts. So what's happening in this little window of time, and this is time in milliseconds, right? Short period of time. What's happening in this latent period? This is when the calcium has to go in, right? The calcium has to go in and attach to the troponin and shift the tropomyosin out of the way. The myosin heads have to bend, and then we get contraction. And once the contraction is over, we get relaxation. That's a muscle twitch. That's a muscle twitch. And the muscle twitch is fastest for skeletal muscle, then cardiac, then smooth. Okay, so the definition here: response of a muscle to a single brief stimulus. The muscle is going to contract and then it will relax, and that's going to happen quickly. Okay. Another definition: muscle twitch is a mechanical response of a muscle cell, a motor unit, or entire muscle to a single stimulation or action potential. Action potential is that full process of stimulation, contraction, and then relaxation. a change in the resting membrane potential, the charge across the cell membrane. Okay. So there are fast fibers and slower fibers. So this is giving you an example of that. I'll zoom in on it. So extraocular muscle, these are muscles around the eye, 7 to 8 milliseconds. Now these are all skeletal, right? So these are all considered faster twitch. We're talking about milliseconds, from maybe 20 milliseconds to 200 milliseconds. Milliseconds, right? Short time we're talking about. So we have all the same amount of latent period where we actually have to get our calcium coming in and our calcium binding to the troponin. The eye muscles, only 7 to 8 milliseconds for the whole muscle twitch. Gastrocnemius, that's the muscle, calf, right? About 40 milliseconds for the whole stimulation, and then recovery, another 40 milliseconds. So maybe extraocular was 4 and 4, no, 8, 7 and 7. 7 for the stimulation, 7 for recovery. Okay. And then the soleus, about 90 seconds for the stimulus. The soleus is the muscle under the gastrocnemius or deep to it, and then another 90 for recovery. So these are fractions of a second. Okay. So fast twitch fibers contract quickly. Slow twitch fibers contract slowly in comparison. Okay. So this is how fast we get that cross bridge cycling happening. These myosin heads binding to the actin binding sites and contracting. Okay. The fast muscles are better able to break down that ATP, and the slower ones are less efficient. But again, we're talking about fractions of a second. TREP or TREP-A, we hadn't talked about that. This is where I keep stimulating the fiber. So this is a stimulus, this is a stimulus. And every time I stimulate them at regular intervals back to back, I get an increase of the force of contraction. So you see that happening here. So when I get I stimulate here at each arrow, and then this is within regular intervals of time, and I get full recovery, but I stimulate it again immediately. And each time I get a stronger and stronger contraction until I get to a plateau where that contraction is maxed out for that particular muscle. Okay. So this represents my muscle twitch. Summation, before I can get full relaxation, I stimulate it again, and then I get a stronger contraction. Incomplete tetanus, I'm stimulating it very quickly, and I'm getting to the point where I'm maxed out, but it's not fully maxed out. When I want to max it out, I have a complete fusion of all of the contractions because I don't let up on the stimulus. There's no delay between each stimulus. And so then I end up with this maximum contraction that's sustained but weak. Okay. So fatigue is where I keep stimulating, and I keep maxing out my contractions, right? So I might have some delay in between each one, but I keep in a regular interval, keep stimulating, keep contracting, and then I get to the point where I've done it so much that I've used up all my ATP, and now I'm fatigued. I cannot stimulate a contraction anymore because I got to build up some ATP. Even though I keep providing a stimulus, I cannot contract because I don't have the ATP that allows me to do it. So that is fatigue. And so this is just showing you that if I have tetanus, that will lead to fatigue. And if I don't have a long enough rest period in between points of fatigue, I'll fatigue much more quickly the next time. And so you see, I don't have that same length of time here that I can sustain a contraction because I needed a longer rest period between one fatiguing sustained stimulus and another. Tone, we defined that already. Recruitment, we've defined that already as well. Okay, types of muscle contraction. First of all, you need to know that the muscles are attached at 2 points, an origin and an insertion. So the origin is the least movable point of attachment or the immovable point of attachment, and the insertion is the most movable point of attachment. So when a muscle contracts, the insertion moves toward the origin. Typically, the origin is more proximal and the insertion is more distal, and so the distal end will move towards the proximal end. So like when you bend your elbow, right? When you bend your elbow, the I think I have an illustration of it. The origin is higher up on the humerus, the insertion is further down on the other on the radius, and when you contract, you move the insertion towards the origin. So we have some types of muscle movement: flexion and extension. Flexion is decreasing the angle of a joint. So when you make a muscle, if you will, extension is increasing the angle of a joint. Abduction and adduction. Abduction is moving the limb away from the body. So if you were standing straight and you just lifted your arms out straight, that would be abduction. And then if you move them back towards the sides of your body, that would be adduction. Rotation and circumduction. If you stick your arms out and you move them in great big circles, that's rotation. Movement around the longitudinal axis of ball and socket joint. So you make big circles with your arms. That's rotation. Circumduction is you stick your arms out straight to your sides and you keep your shoulders still and only turn the distal ends of your arms in circular motion. That's circumduction. Pronation, supination. Pronation, palms down. Supination, palms up. Inversion and eversion. Inversion is turning the foot so that the sole of your foot faces inward or medially. And eversion is turning your foot so that the sole faces laterally or outward. We sit with our feet inverted all the time. Like if you cross your feet or whatever. Eversion, we don't do that too much. Sometimes I have shoes that high heel shoes that have buckles on the outside, and I have to kind of do some eversion to get to that buckle my shoe around the ankle. Dorsal flexion and plantar flexion. Dorsal flexion, moving the ankle so that the toes are pointed up. Plantar flexion, moving the ankle so that the toes are pointed down. Okay, so I did not have the picture that I thought for origin and insertion. Let me see something. Okay, I wanted to find origin and insertion before I talk about this. So let me see if I can share my screen. Okay, I hope that you can see that. There's a picture here showing you the origin and the insertion. And so we have the points of origin more proximally at the top where you see origin of biceps brachii. And then we have the insertion down here on the radius. Okay, and so the attachments at the top are not going to move. When the muscle contracts, the insertion moves toward the origin. Okay. Okay. So we have muscles that are responsible for a particular movement. Those are referred to as the agonist or the prime mover for that particular movement. So when we talk about bending the elbow, the biceps brachii is the prime mover for that particular muscle contraction. Okay. It's the agonist. The antagonist opposes the movement of the agonist. In this case, that would be the triceps. Now, when we say it opposes it, it doesn't stop the movement, it really stabilizes the movement. If I didn't have any muscle activity on this side, then this would just throw my whole arm up, right? So I have that antagonist to stabilize the movement of that area, right? To stabilize the contraction here. Then we have synergists that help the prime mover, and we have fixators that stabilize the origin. Okay, and so we have the brachioradialis that is helping, and then we have the deltoid that is a stabilizer here, for example. Now, again, whether something is an agonist or an antagonist depends on the movement. So if I were extending my arm, the triceps would be the agonist and the biceps would be the antagonist if I were extending the elbow. Okay, so it depends on the particular movement. We name muscles based on a number of things: direction of the fibers, like external abdominal oblique, right? So those fibers run obliquely. It also, we can name them based on the size: gluteus maximus, gluteus minimus, gluteus medius, pectoralis major, pectoralis minor, right? So the size of the muscle, location of the muscle: frontalis muscle, occipitalis muscle, temporalis muscle, external abdominal oblique, right? We named it because it's oblique, but it's the external abdominal layer, right? Number of origins: quadriceps, triceps, biceps. Location of origin insertion, so the points of attachment: sternocleidomastoid. The sternocleidomastoid attaches to the sternum, it attaches to the clavicle and the mastoid process of the temporal bone. Sternocleidomastoid, those are the points of attachment. Shape of the muscle: trapezius. Action of the muscle: so we got flexor carpi radialis, flexor carpi ulnaris, extensor carpi ulnaris, extensor carpi radialis. Those give you an idea of what they do. They flex or extend, and then it also gives you a location, right? Against the ulnar, against the radius. So we have these characteristics for the naming of muscles. Okay, smooth muscle contraction. We're just going to talk about this briefly. No actin and myosin arranged in a way that gives us striations, but we do have actin and myosin. And the actin and myosin has kind of this crisscross shape across the smooth muscle. Now I want to zoom in on this picture because there's a couple of things I want you to pay attention to for smooth muscle. One, we got gap junctions. So when I have another cell against this muscle cell, so I'll have another muscle cell here, right? And then you got some here, right? And so these gap junctions are connections between these other cells, right? But in addition, I have these little depressions called calveoli. These are invaginations or depressions in the cell membrane. Remember when we talked about endocytosis and exocytosis? Endocytosis, we have these little depressions in the cell membrane and they can bring in fluid, right? Penocytosis is cell drinking. These smooth muscle cells have that. And inside of these calveoli or little depressions, we have receptors specific for calcium. So again, calcium is important for muscle contraction. But in the case of smooth muscle, I don't have that sarcoplasmic reticulum. I bring it in from the outside in those little vesicles. Okay. So we have these dense bodies connecting these fibrils, the actin and myosin here. And then this is a contracted smooth muscle fiber. Okay, so for smooth muscle, you don't have the striations, but you have the actin and myosin. They form those little networks that kind of look like a net that we just saw. And they connected the dense bodies. So the dense bodies kind of act like the Z lines, and they hold those stem filaments in place. You need the calcium from outside, so you get it in those caveoli. And that calcium comes in and it binds to a molecule called calmodulin. And when it binds to that calmodulin, it forms a calcium calmodulin complex. And that's going to activate a specific enzyme that is going to trigger myosin and activate the myosin heads. And they're going to attach to the binding sites, and then you get the contraction. Okay, so we don't have the sliding filament happening, but we have a slight twisting and shortening of the muscle that happens. Okay, and as long as we need the muscle contraction, calcium is going to be coming in, and once the contraction is done, we release that calcium. Okay, I'm going to give you some time, and you can always pause this to write this down. I don't think this part is in your notes. It may or may not be, but take some time, if it's not in your notes, to write this down. And you can always pause it. So here's what that looks like. You have your smooth muscle fibers, and then you got the actin and myosin here. And then if you, let's zoom in. So you see these little areas where the actin and myosin interact and cause the shortening. Same thing here. And that is it. Please, please, please write down any questions that you have and email me. If you want to chat, we can chat. We can set up a time to get together and chat about it. I know this is probably the heaviest amount of physiology we have had for this semester so far. We'll have a little bit with nerve as well, for how an impulse is passed down a nerve. So muscle contraction is the most physiology that we've had. We had a little bit about bone production and repair. Okay, so these two sections are going to require a little bit more time put in studying. It may require you asking some questions. Please do not hesitate when it comes to asking the questions. I'm also going to post a video of the sliding filament mechanism as well. So you'll have that, and maybe looking at it actually happening will help you. Okay.